Welcome to an overview video for the theory, the structure of the lab. We will spend this semester about 85% of this semester working on bacteria, how to grow them, how to identify them, with the same kind of structure that one would use if you worked in a clinical lab. So you get a sample from a patient and your job is to identify what it is that's making that patient sick. We'll spend the end of this semester doing a project with protists and fungi and worms. We're not going to work in the lab with viruses because they're so small. They require very expensive specialized equipment that requires extensive training in order to use. And so it's just not feasible to do that this semester. In other classes, in more advanced classes, you may get to do some virology work. But we're going to spend most of our time working with bacteria. So you get a sample from a patient, and the first thing you need to do is to grow up more of that organism so that you have plenty of it to work with. So the first thing that you typically do is to put that organism on in media, or growing it on a solid surface of media. Remember, media is just a way of delivering all the food and nutrients the organism needs. And then you put it in the proper environmental conditions to multiply the organism. Here's an example of the bacteria spread across a plate, which is a typical media choice to multiply your organism. So what's happened here is the bacteria have been spread evenly across the plate to the point that individual bacterial cells have separated from each other, and then those bacteria start copying themselves. They copy themselves tens of thousands, even hundreds of thousands of times, to the point that you can actually see them without a microscope. So these piles, these circles, represent what are called a colony, and that is a pile of identical bacteria that have just copied themselves so many times that you can see them. Interesting thing with colonies is that they're distinct for different organisms. E. coli's colony has a distinct color and shape and texture that's different than Staphylococcus, that's different than Streptococcus. So with experience, the way that they actually look on the plate, called the colony morphology, can give you clues to help you eliminate all the different options out there to figure out what it is that is causing your patient illness. Once you've grown up the bacteria, you take a few of the bacteria from these colonies and you put it on a microscope slide, and you look at what the cells, the individual bacteria, look like on their own. So this is called cell morphology, and this will be the second thing that we do. After we finish looking at what the bacteria look like on the plates, we'll shift over to what they look like under the microscope. There are three primary shapes to bacteria. There are other more minor ones, but the primary ones are circles, which are called cocci, or rod shapes, which are called bacilli, or spirals, which are called spirochetes, or sometimes you'll hear them called spirillum. So most bacteria are cocci or rods. They're cocci or bacilli. Spirochetes are much, much rarer in nature and clinically. Beyond the basic shape of the bacteria, you then look at how they orient themselves with respect to each other. So if the bacteria is completely isolated on its own, they're referred to as a single arrangement. If the bacteria tend to pair up, then they're called diplo. If the bacteria cluster up in big clumps, they're called staphylo. If the bacteria form long chains, they're called strepto, etc. So you'll learn a variety of different arrangement vocabulary, so that when you look under the microscope, you can say what shape it is, and you can say what the arrangement of the bacteria is. And again, this is unique for the bacteria. Escherichia coli has a certain shape and arrangement. Staphylococcus aureus has a certain shape and arrangement. All of that data builds to help you identify what it is that your organism is. Okay? So we're going to go through the most common ones. I do want you to know that in nature there are unique, much more minor examples. We're going to go through the common ones, and those will be the ones that you're going to be responsible for. besides just looking at them under the microscope, we'll also do some staining techniques to look for certain structures, to see if they produce endospores, to see if they have, if they're gram-negative or gram-positive, if they have 2 layers or 3 layers on their cells. There are lots of differential staining methods that you can use to look for certain shapes or certain structures to give you more information about the organism. Once you're finished with looking at it, the next step will look to see what chemical reactions the organism can do. And that will be really about 2/3 of the semester will be spent learning the basic chemical tests that are commonly done to help you identify, because a lot of bacteria are, gram-negative bacilli that have the same basic structures. Well, so what do you do beyond what do they look like? You have to start really digging in deeper, and that's what the biochemical tests will allow you to do. So we'll have our first unknown project that will be, we call it the morphological unknown. It's going to be based mostly on what it looks like, both in colony and cell morphology and the basic structures. And then the second unknown will be, it'll start with what does it look like, and then it'll dive into the biochemical tests, which reactions can it do. And that will really allow you to dig deep from a vast array of bacterial options down to a single one that is your unknown your unknown organism that is infecting your patient. And again, we're going to finish up with a project using some microorganisms. You'll be able to see some fungi and some protists and some worms in the lab, but our emphasis is going to primarily be bacteria. Hopefully you learn lots of amazing things and have a wonderful time this semester.